So good evening, everybody. So Concord Biotech is almost 20 years old organization, which is having its expertise, particularly in global biotechnology and immunosuppression as experts. Moreover, it has started its operation in the year 2000, where Dr. Sudhir Vaid, a renowned biotechnologist expert, started this company with few immunosuppressants. Moreover, in 2005, Concord Biotech got its first US FDA inspection and got its approval for API facility, and then its second approval in the year 2008 for US FDA. In the year to come, it has got many approvals in time to come, and in 2019, it has got approval for US FDA ANDA. So Concord Biotech launched its critical care formulation business in India in that particular year in 2019. So you can see that is having all the approvals like US FDA, European Union, GMP, all the other approvals that are required to have the business, particularly for API and also in finished formulation. So it is having two plants based at Ahmedabad, one in Valthera and one in Dholka, both near Ahmedabad, for its formulation facility and also for API facility. It is having the US FDA approval for both the plants, along with European Union GMP, Korean GMP, and TCT approval. It is having its presence in more than 50 countries globally, like in European countries like UK, Germany, France, Spain, in Middle East countries and Africa like Turkey, Jordan, Iran, Tunisia. More than it is having its presence in North American and Latin American countries like UK, Canada, Colombia, Venezuela, Argentina, Brazil, and it is having the vast presentation, particularly in Asian countries like Japan, like China, Korea, Ukraine, Australia, all these countries having its presence. So you can see that Concord Biotech is having its global presence, not only in India, but in other countries, particularly its expertise mainly in API formulation. So for the next slide, you can see its presence in globally for immunosuppressants range. Like for tacrolimus, for Tacrocord, 1 out of 2 patients globally are on Concord Biotech tacrolimus, particularly for its API. Similarly, for mycophenolate sodium, 1 patient out of 5 patients are on Concord Biotech API. Similarly, for mycophenolate mofetil, 1 out of 7 patients, and for cyclosporin, 1 out of 10 patients are on our raw material. So more than 5,000 patients has been enrolled so far on Concord Biotech immunosuppressants, and many more are counting with time to come. Concord Biotech is having its presence in European and US market, as you can see by the studies of BABE, which is very much required for the approval of US FDA and European Union, where we have compared our molecule with that of the innovators. That is, you can see for Moficon S, like Tacrocord. So you can see the graph is almost matching for tacrolimus and also for mycophenolate mofetil and sodium. So you can say that our drug's efficacy is almost equivalent to that of the innovator. Similarly, we are having our state-of-art R&D facility based at Ahmedabad, where our lot of scientists are working for the R&D and working for the benefit of the mankind in time to come, so that they can provide us with the medicines like for oncology, immunosuppressants, or antibiotic and antifungals. Similarly, as we mentioned in earlier slide, that we are having state-of-art 2 sites for your formulation and finished formulation. A new plant is coming nowadays in time to come just near Baroda, where we will again have the second API plant in 2 to 3 years of time, with all the approvals in place. So presenting now the INCA, that is Intensive Care Division of Concord Biotech, particularly for critical care patients, those who are suffering particularly in ICU and very acute type of illness, to provide global quality and saving precious lives in time to come. As Concord Biotech is providing API, particularly for products like Muprosin, calcium, then Teicoplanin, Daptomycin, Phosphomycin, Vancomycin, Polymyxin B, then your antifungal like Micafungin, sodium, we are providing the raw material. So our raw materials expertise is that for the fermentation process, we are not exporting the raw material from the China or chemically synthesis process. We are particularly using the bacteria that creates the raw material, and that raw material is the pure form of raw material, and that is going to have the edge over the chemically synthesis raw material. As you can see, that is why we got the approval of US FDA, European Union, and other approvals. Our upcoming APIs are that for tobramycin, then your retampulin, then your telavancin, then your antifungals like amphotericin B, then your caspofungin. All these APIs that is going to come in time to come. Apart from that, we are having our own API manufactured products like Anicod that contains anidulafungin, micacod, micafungin, ticoplanin, 200 and 400 dapute, that is daptomycin, Pobix, that is polymyxin B, 5 lakhs international units, Phosphotrac, that is phosphomycin for injection, Phosphotrac 3 gram sachet we are also having, then your Vanogard 500 and 1 gram of vancomycin, Muprovant, that is muprosin ointment, and very soon we are coming with amphotericin B liposomal, 50 milligram. Similarly, we are redefining quality and care with extended portfolio like Caspocon, Borexia for antifungals. In antibiotic, we are having Mapicon, Mapicon SB containing meropenem and salbactam, then Cricolist containing colistin, Nexmatch 4, Minocrit, Primitaz, and Tigicon. For the affordability of the patient in today's market, where a lot of patients can't afford the very costly antibiotics, we have launched the EVA range, where E stands for effective infection control, V for versatile action, and affordable price. Similar quality that we are offering for like Caspo Con, we are offering the similar quality for Caspo EVA with the very reduced price for the benefit of the patients and also for the hospital's patient benefit. Like Caspo EVA, Mero EVA, Hope EVA, Imp EVA, and Imp EVA Fort. We are also, Concord Biotech is also associated with CSR activities where we are providing the educational scholarship for the very needed person and those who are having very bright mind. Similarly, we are providing the mobile health checkup van, then your environmental protection. We are doing a lot of things, particularly in Gujarat and other states also. We are contributing in water harvesting. Similarly, we are regularly participating in blood donation camp all over India. So thank you so much for my small presentation. And with that, I am inviting Dr. Inderjit Tiwari for his presentation. And in the end, we will have the question answer session where we will have question for Dr. Tiwari from the audience. After that, we will end our presentation. Thank you so much. And over to Dr. Tiwari.
Yeah, thank you, Mr. Amit. It's a nice so you have given out the outline of your company, and I'm glad that you are covering the most of the antibacterial and antifungal as well. So basically, most of the intensivists may not be aware of your all the products, but I think since we have the whole basket of product, almost we are covering critical care products like antibacterial, antifungal. I think there is a opportunity to serve it better. And I am really impressed with your last part, that is low cost antibiotics and antifungal you are launching there, or you are making it available for low cost for the hospitals, even for lower cost hospital. That is a great thing. That is a great thing. I must appreciate. So now I will share my presentation there. Can I share it?
Yes, sir. Yes, sure. Sir, it's not visible.
I am just because just you have stopped sharing, so I have to reload the thing.
Sure, sir.
Give me just 1 minute time, so I have to reload it again. Or reload the file. Yeah, are you getting it?
Yes, sir, it's visible. Just make it. Yes, sir. Yes.
Again and again, getting the same problem. Reload the file. 30-second more. I am visible there.
Yes, sir. Yes.
Yeah, my slides are visible.
Yes.
Okay,
Right, sir.
thank you. Yeah, now I am starting my presentation. Today's topic is role of echinocandin in ICU. We'll be wondering that why I have narrowed down this spectrum, because we are not discussing the role of antifungal in critical care. I have narrowed down is to the echinocandin. So first question in your mind must be why echinocandins? So because of the two regions, echinocandins now it is the most preferred and most efficacious drug because of its safety profile and broad spectrum coverage in the antifungal arena. So that is the one point. And second point is that in critically ill patient, uh, basically this is the drug of choice in most of the scenario. If you are considering candida as per se, basically if you are considering the candida in ICU setup. So because of the two regions, the broad spectrum activity against the candida and other fungus. Second is the safety profile, and third is the choice because of this availability nowadays. Because few years back it was not available so frequently, so it is one of the highest used antifungal in the critical care, and think that it needs a discussion for 30-35 minutes. So invasive fungal infection, the revised definition, that is, we particularly divide into three groups. One is the proven case, one is the probable case, one is the possible case. So proven case are those which demonstration of fungal element in the disease tissue for most condition in patient that is identified a culture and indirect assay. That means these are the cases. Proven cases are basically culture positive cases. But I must tell you, this culture positive reports, you should not wait for this culture positive report, because you have to start as a presumptive treatment in case of probable cases, or even you have to give prophylaxis in your high risk cases. Otherwise, even after a culture sensitivity report, if you are starting your treatment, antifungal treatment, the mortality is as high as 40 to 50%. That means if you are starting it or you are waiting for the culture report, your mortality, even after the treatment, full treatment, sensitivity treatment, the mortality is almost 40 to 50%. So our main idea is to give the preemptive treatment, empirical treatment, or the prophylaxis treatment. Otherwise, once it is proven, the mortality is very high. So the probable cases will be demonstration of the host factor, clinical features, and mycological evidence in suspected patient. That means you are suspecting some patient with some clinical feature. Suppose any patient is having on higher generation antibiotic, still patient is febrile, procal is normal, and or candida score is high. In that cases, these are the probable cases, and you must start the empirical treatment. I think this is the subset of population which is the target on radar. So this should be the target population where we can start it empirically so that we get a better result. I will later on discuss how the time lapse which affect your patient management. Starting it early is always a best option in any interfungal treatment. And third group is possible invasive fungal disease. That means demonstration of the host factor and clinical feature in suspected patient without any definitive mycological evidence. That means these are the cases in which there are host factor. That means high risk cases, clinical features are also there in suspected patient, but without any definitive mycological evidence. In that case, serum biomarkers are also negative, but you think these are the high risk cases. Suppose any post liver transplant patient or any patient having big surgery or prolonged ITU stay. In these cases, we give the prophylaxis. So these are the basic three groups. In the pathogenesis, transmission mainly via the disrupted anatomical barrier of the body. Vertical transmission from mother to baby is also recorded. Horizontal transmission, patient to patient, contaminated infected health worker or hospital acquired. Basically, the uh, hand washing. Hand washing is one of the things which is very important because patient to patient. Basically, if you are observing the trend in last 1 year, not 1 year, basically in last few years, there is a sporadic cases which become epidemic, particularly in case of Candida auris or Hemolyne. If your one patient is getting this auris or Hemolyne, ultimately you are landing with 3 or 4% consequently. Why? Because of this patient to patient or healthcare worker or hospital acquired infection. So that is one of the important concern, and only option is hand hygiene. Second thing is the colonization. Colonization, the sites, you have to locate the sites because you must have to find out the what is the colonization index in particular cases so that you can start it empirically or you can start a prophylaxis study. So colonization site you have to look for, that is skin, respiratory tract, GI tract, central vascular catheter, or any invasive lines. You should look for the colonization of this candida, not candida, any fungal infection. And the other risk factor, that is patient, like geriatric patient, diabetic patient, immunocompromised patient, organisms, and drug related. And third thing is infection, clinically manifested disease. One important finding is that different studies show that sometimes this candida infection, basically candidemia, is related with bacteremia. In 40 to 50% of the cases, underlying bacteremia may be there in occurrence with this antifungal. So you have to keep in mind when you are considering the pathogenesis of invasive fungal infection. So the etiological part, the immunocompromised host or high-risk group, transmission via the broken mucosal barrier, and presence of opportunistic fungi in the ICU. So these are the basic and pertinent risk factor because you cannot modify the host factor or high risk factor. Transmission via the broken mucosal barrier in postoperative cases in bigger surgeries, and that is the presence of opportunistic fungi in ICU. It is always there and they always threaten, always a risk for the immunocompromised patient. So two things we have to keep in mind, that is colonization. Colonization is the development of a fungal infestation in individual as demonstrated by a positive culture. The infected person may have no sign or symptoms of the infection while still having the potential to infect the other. So it is very important because in colonization, your patient may be asymptomatic, but that patient may be serving as a source for infection to the others. And with the same organism, other immunocompromised patient or delicate patient may become symptomatic. So colonization is one of the important factor. And invasion is the fungal invasion or uh, mycological development and spread to the infectious disease, and the infected person have the definitive sign and symptoms of infection. So difference between this colonization and invasion is that in colonization, the organism is not producing any sign or symptoms, and in invasion there is definitive sign or symptoms of the infection of this fungi. So we have to make it clear that difference between the colonization and invasion, because in most of the colonizing cases, colonizers we do not treat, but in sometimes we have to treat it for the benefit of the patient per se and as well as for the benefit of the other patient. So the fungal infection a real problem in ICU? Yes. Troublesome fact about the invasive fungal infection, high rate of incidence, global and Indian, I will later on discuss. Now it is an emerging concern in critical care area. Why? Because the longevity of the patient, critically ill patient like advanced malignancy, immunocompromised patient, retrovirus positive. So all these patients are living more with the advancement of the therapy in case of chemotherapy or immunomodulation drugs. In that case, there is always a chance of more opportunistic infection, including this fungal infection. High rate of incidence, high rate of mortality and morbidity, as I have told you, that the mortality is much higher than bacterial or viral infection. Even in culture positive cases, mortality is as high as 40 to 50%. And most important thing is it is very difficult to diagnose because culture positivity is almost 50 to 60% cases it is culture positive. That means your suspicion empirical therapy, and the monitoring of the treatment parameter. These are the main things you can do when you are considering this fungal infection. Global burden, you can see the number of infection per underlying disorder per year, like in HIV, AIDS, respiratory cancer patient. If you're taking this ICU patient burdens like Candida peritonitis, invasive aspergillosis, and total burden estimated, it is also very high in ICU patients. Why India has a high risk for the invasive fungal infection? Due to few reasons. One thing is overuse of antibiotics or antimicrobial drugs. Yes, it is one of the important aspects when you are considering this, the incidence and prevalence of Candida infection. Sometimes our antibiotic policy in most of the hospital is not regulated. So sometimes overuse, or I must say the broad spectrum antibiotic. This is a culture to use a broad spectrum antibiotic and high generation antibiotic in most of the critical care areas. So it is one of the important facts when we are talking about the invasive fungal infection. Second thing is diverse climatic condition, heavy annual monsoon. This is per se a concern due to the climatic variation, the environmental factors, the growth of fungus, and this is a favorable condition which is there in the Indian subcontinent. Very few good diagnostic mycology labs. So most of the laboratory, I must say, very few laboratory has the facility to identify the subspecies. So you can identify albicans, non-albicans, but after that, sub-specification or drug susceptibility testing are very available in few centers in India. And lack of credible Indian data on incidence of invasive fungal infection. Yeah, this is also one of the important thing, because sometimes you unable to report the case, you don't have any robust data to represent the scenario of all over the country. These are the things which makes us the high risk for the invasive fungal infection. If you compare the trend comparison between the Candida trends, India and Western countries, so in India, it is the third most common nosocomial infection, and in Western country, it is the fourth most common nosocomial infection. In India, Candida non-albicans is most common. In the Western country, still it is Candida albicans. If you consider the species, Tropicalis in India, Tropicalis is the highest prevalence, followed by albicans and parapsilosis. Whereas in Western countries, albicans still is more than the non-albicans, and among the non-albicans, glabrata and parapsilosis are there. And overall incidence in Indian scenario is 6.5 cases per 1,000 ICU admission, whereas in the Western country it is 3.6 cases per 1,000 ICU admissions. So that means we are far ahead from the Western countries, and we are really concerned with the prevalence of more and more prevalence of these non-albicans cases, which are some of these species are very difficult to treat. So if you look in this graph, the Candida trends in the ICU, Indian ICU, that means rapidly increasing rates of incidence in last decade. That is overall range from it was 17.7%. Now it has been very much raised. You can see the graph. This is the albican cases, and this is the non-albican cases. So the rate of candidemia in the ICU over the study period, that was between around 2012, it was almost 29.5%, which was very much significant. Okay. Now the switch from the albicans to non-albicans, as I have told you, the notice changing trend of the Candida albicans to the non-albicans species in Indian ICU. So it is drastically rising. Initially, if you look after 2001, 2, it is almost going parallel. But after 2003, 4, there is a spike in the cases of non-albicans. So the important finding by, if you consider this study by Singh et al., 2014 study, Candida is the most common invasive fungi in India, that is 93.4%, followed by Aspergillus flavor. That means if you are considering the overall fungal situation, Candida, Candida, and Candida, because it is 93.4%, and Aspergillus is 3.3%. Among the Candida, most common isolate is non-albicans, and it is almost 71.8%, followed by the albicans. So this change in trend I have shown you in the last two graphs, that it is acute. That means in last 1 decade, that rate has been phenomenally increased. And among the non-albican Candida species, Candida tropicalis was the most common isolates. And this ICU acquired candidemia in India, if you look after this study by Professor Ronaluk Chakravarti, the fungal man of India, in the study was done in 2015, that was a very good prospective multicentric observational study conducted in 27 ICUs across the country. And burden of ICU acquired candidemia is reported to be 6.51 cases per 1,000 ICU admission, and burden high in the north 8.95 and lowest in the west 3.6. And mortality, majority of the patient are non-neutropenic, so we should not be in an idea that neutropenic patient has more in number. No, in majority of the patient were non-neutropenic, that is 98.7% in this study, and medium duration of onset of candidemia was 8 days. So non-albicans infection was 79.1%, whereas the albicans was around 21%. So in India, as I have discussed, is the tropicalis most common in 41% of the cases, albicans 20%, parapsilus is 10%, and gilberata 7%. In this particular study, you can see this majority 42% is tropicalis, followed by this albicans 21%. In developed world, Candida albicans is still the most commonest, that is 45 to 70% of the cases, followed by gilberata and tropicalis. And mean age of candidemia patient was 49.7 years, around 50 years, and have lower Apache 2 score at the time of admission. Candida tropicalis more in private or corporate hospital, whereas Candida auris and Candida rugosa in public sector. But this is one of the important finding, because I've told you these are the notorious bug like auris, hemolini, rugosa. Once you get it in your setup, it is going like anything and makes a focal zone of epidemic. So 6.6% of the isolates were most of the isolates were susceptible to all antifungals. In India, the species specific resistant rate against the fluconazole was 5.2%, albicans 4%. It was 5.2% in Candida albicans, 4% in Candida parapsilosis, 2.6% in tropicalis, 1.5% in gilberata. If you look up to the overall resistant pattern of like fluconazole is around 10%. Now it which increase. So species wise it may vary, but overall it is seen up to 10% of the resistant with fluconazole in different setup. Among types of surgery, abdominal surgery is considered a leading risk factor for candidemia, and removal of central venous catheters and start of targeted antifungal therapy within 48 hours of onset of candidemia improve the 30-day survival in the patient. The 30 days COVID mortality is similar to the EPIC 2 international series. That is, I have already told, it is between 40 to 50%. In EPIC study was 42.6. In this study, 44.7. It is around 40 to 50% the mortality reported. Now, it is very important slide, relationship between the mortality and timing of antifungal treatment. The rate of mortality is higher in cases in which antifungal treatment is delayed for more than 12 hours. So every hour matters. I must say this should be treated as a golden hours. So if you are making it delay even by 12 hours, your outcome varies. And if you are delaying more than 48 hours, definitely there is a significant difference in outcome, both in case of mortality as well as in morbidity. So again and again, I am emphasizing the early treatment, that is empirical treatment, or even in the we can think for the prophylaxis. And principle is same in this case also. You have to strike it hard like antibacterial. You have to choose the most potent antifungal. Later on, you can de-escalate according to the clinical condition and some biomarkers. So delay in the antifungal therapy and hospital mortality. Similarly, they to start in fluconazole on the cultured day or more than 3 days. So every hour of delay, every 12 hours of delay makes a difference in the outcome. The risk factors we have discussed, that is organism, patient and drugs, 3 different risk factors. Organism, it depends upon the virulence property, number of organisms and multiple sites of colonization and adherence properties. For the patient profile, patient related, immature or inefficient immune system, moist skin surface, mucosal breakdown, invasive catheters or any lines, tubes or central venous lines, and drug related, like if you are using antibiotics for more than 2 antibiotics or patient is on multiple antibiotics, H2 antagonist, postnatal steroids, parenteral nutrition, lipid emulsion. A lot of more factors are there, but these are the some common factors you have to keep in mind. So, if you have to summarize in this risk factor part, if your patient is having more than 3 antibiotics, antibiotic used for more than 4 days, more than 4 days of ICU stay, mechanical ventilation for more than 48 hours, major abdominal surgery, CVC or any other invasive lines, total parenteral nutrition, neutropenic patient or immunosuppressive patient or patient on immunosuppressive drugs, concomitant infections, diabetes mellitus, candida colonization at more than 2 sites, and candiduria, that is more than 10 to the power 6 colonies you are getting. Then, will I start an antifungal in my unit? This is a very important question because every intensivist want to know when to start this antifungal prophylaxis, empirical and definitive. Definitive, you have the ready-made answer, you get a culture positivity, you start according to the sensitivity, but you don't have any price for that. So thing is that start empirically or the prophylaxis. So in prophylaxis case, you have to choose the high risk cases, and according to the high risk cases, candida score and your clinical suspicion, you have to start the prophylaxis. But most important is the empirical therapy, when you will start it. So new unexplained infection already on antibiotic, as I have told you, patient running on fever with your high generation antibiotics, you must think for your antifungal. Multiple site colonization, long stay in ICU patient, immunosuppressed or vascular access or an invasive catheters are there. Now, part, as I am repeatedly discussing it, prophylaxis, empirical, preemptive, and targeted therapy. Prophylaxis, that is, patients do not have any infection, but they are the high risk group without any clinical symptoms of infection. Empirical, that is the major thing where you have to start it, the treatment. These are the possible infection, high risk group with pertinent persistent fever despite of the antibiotic therapy. Now the probable infection, where you are suspecting it more likely but unable to prove it because of culture negativity. So these are the high index for suspicion but no confirmed diagnostic proof. And last part is target therapy, when proven infection, full-blown disease or organism is identified. So on the treatment part, if you consider this evolution of antifungal therapy, treatment of choice and choice of treatment, that means it is evolving from the azoles, from the echinocandin and the liposomal preparations. So spectrum of, so I am not discussing individual like azoles or echinocandins or like pollines. So I will focus the talk on this echinocandin only. So spectrum of infection of the systemic antifungal agent for the candidiasis. You can see this pollens, triazoles, echinocandins and others. So this fungal genera whose resistant is possible, that is 5-fluorescein group, and other most of the part are sensitive, except this Candida glabrata. These are intrinsically resistant to this azoles. Triazoles should not be given in case of glabrata. Like this spectrum of answer of action of the systemic antifungal agents for the molds. So most of them are sensitive with the triazoles, and with echinocandin, two species, that is Aspergillus fumigatus and Terrius, and most of the study with caspofungin look sensitive. Otherwise, these triazoles are good with this mold infection. If you consider this echinocandin mechanism of action, this acts on the cell wall and it inhibits the glucan synthesis. It is a glucan synthase inhibitor, and it will cause the inhibition of the beta-1,3 glucan synthesis. So by this mechanism, cell wall synthesis is disrupted and it works. Mechanism of action is to the inhibition of beta-1,3 glucan synthesis. And why echinocandin? So I will show you 2 or 3 study which will conclude that this is the drug of choice in critical care because of 2 things. One, in critical care, as I have discussed, most of the species is Candida, and Aspergillosis is in only 3.6% of the cases. So if it is Candida, then our choice is between the azoles and echinocandins, and critically ill patient, the echinocandin is favored, and now it is a drug of choice by the IDSA or Defense Society. So why echinocandins? 1 or 2 study I will quote there. This is a multi-center randomized double-blind phase 3 non-inferiority study of echinocandin versus fluconazole for the treatment of invasive candidiasis. In that case, you can see there's clinical success, that is signs, symptoms of invasive candidiasis resolved, no additional systemic antifungal needed, and the microbial success. It is basically difference is very significant, like 80 to 96% in comparison to 50 to 71% in case of fluconazole. So global success is quite significant. Individual patient level quantitative review of 7 randomized double-blind trials for treatment of invasive candidiasis. Total number of patients enrolled was 1915. And study drugs represented 3 different classes of treatment, that is liposomal amphotericin, anidulafungin, caspofungin, micafungin, fluconazole, and voriconazole. So the treatment success rate, overall mortality was 31.4%, and one of the important factor in this 30-day all-cause mortality was echinocandin treatment. The p-value was significant. That means this is one of the important part, along with this routine things like removal of the catheter, Candida tropicalis, which is more common, immunosuppressive therapy, higher Apache score increasing is. So this is one of the important aspect they have evaluated in the study. And the IDSA guideline on the initial treatment of candidemia in non-neutropenic patient. So I am basically narrowed down to the clinical aspect only. I am not covering the whole big spectrum of these things. So in non-neutropenic patient, drug of choice is echinocandin in critically ill patient recommended. Dose is Caspo is 70 milligram loading followed by 50 milligram per day. Mica do not need any loading dose, 100 milligram daily. Anidulafungin loading dose 200 milligram, then followed by 100 milligram per day. So the alternatives are there with fluconazole, liposomal amphotericin, and voriconazole. Fluconazole usually we used as a basically in critically ill patient. So like antibiotic stewardship. Similarly, we do the on fungal treatment, antifungal stewardship. So we narrow down the spectrum. On once become patient stable, we can shift to the azoles from the echinocandin. Liposomal amphotericin B, that is reasonable in cases of intolerance, limited ability of other antifungal if there, and recommended for suspected azole and echinocandin resistant candida infections. So this is also limited role. And voriconazole effective but little advantage over fluconazole. In this active study, that is they have compared this isobuconazole versus caspofungin for the primary treatment of invasive candidiasis, and basically they concluded that isobuconazole didn't meet the criteria for non-inferiority to the caspofungin. Now the breakthrough of invasive fungal infection, micafungin versus fluconazole as a prophylaxis during the neutropenia in HSCT recipient. So this was also a multi-center prospective randomized double-blind phase 3 trial, and they have included 882 patients and given micafungin 50 milligram per day versus fluconazole 400 milligram per day. And primary endpoint was absence of fungal infection through post-treatment on week 4. So 80% in micafungin versus 73.5% in fluconazole with a significant p-value of 0.03. Now the last part of this discussion, that is combination therapy for the invasive fungal infection. Combination of caspofungin and an azole or an amphotericin B formulation in invasive fungal infection. Some case study findings are there. Caspofungin combined with an azole or with amphotericin B is a potential therapy for serious fungal infection after failure of the conventional therapy. So it is not a routine use in case of antifungal. So this is not a favored therapy even, I must say, but the combination therapy in case of fungal treatment is not so common because of few reasons. But in some cases, when you are facing the resistant, basically to treat the resistant bugs, sometimes in, I have told you, hemolinic, glebrata, or this or oris cases, sometimes you need a combination therapy. So the result of this study demonstrated the efficacy of combination of antifungal therapy with 12 successful outcomes. 12 successful, that means 71% of the cases. The overall successful response rate at the end of the combination therapy reached 57%, with the highest rate observed with the combination of caspo and this azole. So notice fall in the survival rate with delayed combination therapy. So if required, you have to start it early, even the combination therapy. So if you compare this caspo, mica, and arugula, so antifungal activity, I must say, is almost same with caspofungin has a edge on aspergillus because it is more being used for a long time, and some case series and reports are there with the efficacy of combination therapy in case of aspergillosis also. Additional approval, that is invasive aspergillosis, I have told you in case of caspofungin, in febrile neutropenic patient, and micafungin is favored in case of prophylaxis in stem cell transplant patient. Loading dose, I have told micafungin not required it, and pediatric patient, the both caspo and mica can be used with good safety margin, and the renal dose adjustment is not required in any of the three. But in hepatic disease, in advanced cirrhosis and very high bilirubin, sometimes it may require a minor dose modification, particularly in case of caspofungin. Thank you very much. Although it's a very narrow down presentation, or it's a narrow spectrum presentation, I must tell, because we have only focused in the role of echinocandin. So I have not much discussed about the diagnostic part or the spectrum of antifungal or spectrum of the fungus itself. So I have limited my talk to the echinocandin. Why echinocandin is important, I have told you, because of his safety profile and potency and broad spectrum antifungal activity. So either of the three is interchangeable except in few cases with some safety profile. And finally, it is the taper up therapy or switch down therapy or stewards should be followed. And last but not the least, that you have to keep in mind the resistant pattern of this echinocandin also, although it is very rare, it is only seen in 2 to 3% of the cases, but still you have to keep in mind because you are now using it left and right in critically ill patient. So I have tried to keep focus on this single topic, the role of echinocandin in ICU. Thank you very much. If you have any specific question, we can discuss. Thank you. Thank you very much.
Thank you, Dr. Tiwari, for such a lucid presentation. I am sure that everybody in the audience must have been benefited with this presentation. So now I am having few of the questions from the audience that I am going to have for you. So out of the following question, first one is, how is your experience with the use of echinocandins, particularly in your ICU, that is in Apollo Clinicals?
Yeah. Basically, we go with the guideline of IDSA guideline. That is, in critically ill patient, as I have told you, by default, is the drug of choice is echinocandin. Not for the treatment, even for the prophylaxis in some high risk group, we can use it as for prophylaxis also. but the policy is same. You strike it hard. You start with the echinocandin. Once your percentage is stable, or you get a culture sensitivity report, you can down a step, or you can make it narrow spectrum towards the antifungal. So our policy is that any hemodynamically unstable patient, critically ill patient, you have to choose. It is always and always it is a echinocandin, because as I have shown in the literature, our concern is always candida, and followed by in subset of population, that is aspergillus. So if you consider this study, 90-95% are candida and candida, and if it is candidemia or candidiasis, all the guidelines suggest the drug of choice is echinocandin. And among the echinocandin, as I have told you, all the three are in most of the cases are interchangeable, except subhepatorenal more safety towards the nidolafungin. So that is our protocol. First thing, choice is echinocandin, strike it hard. You must think for the tapering off or switching to the narrow spectrum. And fourth is, all the fourth are three echinocandins are interchangeable with some hepatorenal safety more with the micafungin. Andrula fungi.
So, sir, my next question from the audience side is that what is the prevalence rate of invasive fungal infection in your hospital and also in your ICU, particularly in gastro
Yeah,
department?
it is. I have told you, like the Professor Anunalok Chakravarti,
Mm.
this study we have also participated, and we are the part of the study in from the Eastern India, and the incidence we have reported is like 6 to 8%. So in critically ill patient, 6 to 8 per thousand ICU admission, not percent, 6 to 8 per thousand patient we have found. So it is same in Indian continent, that is 6 to 8, 6.7 they have shown.
Sir, as you have clearly mentioned that you use echinocandins for invasive fungal infection, but few of the participants also like to know that apart from echinocandins, do you use any other antifungals for invasive fungal infection apart from echinocandins?
Yeah, we use all the three, and you have personally I have used most of them, including from the newer ones. So like most commonly used is still is not a kind of, most commonly used is fluconazole. Fluconazole means azoles. Among the azoles, fluconazole is the most commonly used because of two or three reasons. Because in 90% of the cases, it is still susceptible. Second, it is very cheap, and third is no major side effect as per se. So fluconazole is most commonly used drug, but in critically ill patients, most commonly used drug is inocandim. And liposomal amphotericin B, personally, I have used a lot because of this cryptococcal meningitis and immunocompromised patient. I have used it a lot. But for the aspergillosis part, we have always used this aspergillosis or yeah, a kind of liposomal amphotericin B and voriconazole. So now we are using in some cases the posaconazole, and if once available, ibuconazole we may use in the part. So I have used almost all, but if I have to narrow it down, most commonly used is the fluconazole for the prophylaxis as well as treatment purpose because of its easily availability, low cost, and still it is sensitive in 90% of the cases. But most commonly used in case of hemodynamically stable, what I have used or everybody used in critical care, it is a echinocandin. And in some cases, that is when you have aspergillosis in your mind, particularly in the some lung cases, some aspergillus pneumonia. In that case, liposomal amphotericin we have used, and some combination therapy also we have used in some resistant cases in case of Orich and hemorrhage.
Like you just touched around the resistance. So here is a question from one of the participant that how frequent you have seen the resistance with antifungals
Yeah,
in
that is very important question because whatever the literature is saying that is 10%, around 10% resistant to the azoles like fluconazoles. But in my personal experience, and now you are getting more and more papers with the resistance in echinocandin is also prevailing, and much more what you are getting it in the literature because what we are finding right now we get it published 6 months, one year or 2 years later. So for the clinical part, I am now getting started getting these reports of content resistance also, And particularly, I must say, what I have initially told you, all the three echinocandins are interchangeable, but now we are getting that some species is resistant to echinocandin, suppose caspo and mica, and they are resistant to anidula, or vice versa. But since it most of the use among the echinocandins is a caspofungin, most of the huge molecule because of its earlier availability, and now all the three are equally available. But for the last 5 7 years, if you consider it, it is the caspofungin. So in my experience, one experience, I am getting few cases more frequently now, which are resistant to this caspofungin. And then you have to must keep in mind why this resistant pattern is going on. I must tell you, it is like the antibacterial resistant. If you are not controlling the source of infection, you are going on giving this antifungal like antibacterial, then ultimately that bug becomes resistant to that particular drugs. Post control is still a very important, not still, it is a very important part in the management of this fungal infections.
So you mentioned about the some advantages of Anidula fungin, but
Yeah.
still there are participants who want to know your preferred choice for echinocandins out of three.
Yeah, as I have told you that all the three are interchangeable, depend upon the availability of the products. If all the three are available, then I have to take the hepatorenal safety. If there is a hepatorenal compromise, we usually choose the annular fungin. If there is no hepatorenal issue is there, then either of the three is good enough. And uh, if you are thinking, because sometimes that is the two or three things keep in your mind. Suppose you have you want to start this annular fungin, so you have to start with this loading dose of 200 milligram, followed by 100 and 100, that is fourth dose. And if you are using this micafungin, you don't have to give the loading dose. So single day therapy cost become very high if you are using this fungin. So cost wise it goes very high for first 1 or 2 days. Although the overall 7 10 days treatment cost more or less equal, but if you go for the first 2 days, so it is very difficult to convince the relatives that it is so much costly treatment is going on. So basically the loading dose is one concern you have to keep in mind. Second is the hepatorenal safety, and third is the organism per se. So my choice is like that. Among the echinocandins, if it is hepatorenal profile is there, go for annular fungin. If there is no hepatorenal issues is there, then either of the echinocandin we prefer. And there is no preference for anyone.
So there is a last question from the audience. The question is, in which condition we should interchange among echinocandins?
Yeah, as I have told you, and because the last 1 month I have get two cases when I have already put my patient on Caspo, and that patient was not improving. Not improving means I am not getting the culture negativity. Two subsequent culture become positive. Although it was showing, I don't know, it's showing that's sensitive to caspofungin. But once we change it from the caspo to mica, the culture become negative. So sometimes it is in vitro and in vivo things are different. So that means in clinical basis you can change. And second thing, if you got some culture sensitive report which you are showing that you are ongoing echinocandin is resistant, in that case you have to change to the other one. But the incidence I have told you is very low, only 3 to 5%. So you have to change in only in case of 3 to 5%.
Thank you so much, Dr. Tiwari, for giving all the questions answered. And I'm sure that everybody here must be benefited with your experience, particularly in your ICU setup, as you are practicing there for the long time and treating a lot of serious patients, particularly in your ICU gastroenterology department. So with that, we have come to the end of this wonderful presentation by Dr. Inderjit Kumar Tiwari. And thank you so much, sir, once again for giving such a wonderful and lucid presentation for the benefit of our audience from all over India, particularly from the eastern part of the country. And I'm sure everybody will learn something from this presentation of yours and your experience particularly. And I'm sure everybody will get benefit out of this presentation. Thank you so much, sir, and have a
Thank
nice
you,
evening,
Amit.
sir.
Yeah, thank you. Take care. Goodbye.
Thank you, sir.